in this session, we'll see the enzyme chapter. We have already seen carbohydrates, their metabolism, lipids, their metabolism, then amino acids, proteins, vitamins, and minerals from the biochemistry. And there are only three chapters remaining from biochemistry, and those are nothing but the enzymes, the nucleic acid, and then replication, transcription, translation. These are the five topics remaining from your biochemistry. Rest of the syllabus we have already completed. Okay, so those who are new, they can join us, and they will able to watch all the previous lecture in application. Okay, and those who are already in our batch, they will have idea regarding what is going on. Now today we'll see the enzyme chapter. Again, this chapter is very important for your GPAT as well as Niper, drug inspector, government pharmacist exams, etc., etc. So recently I'm just compiling the drug inspector previous year paper. So I'm finding over there the questions which are from enzyme chapter. So I have already highlighted those questions at a specific points. Don't worry, I will tell you. So biochemistry of enzymes, we'll see. So let's start. So first of all, what do you mean by enzymes? So the term enzyme itself means it is a catalyst, and particularly it is a biocatalyst. So what do you mean by catalyst? Any substances, any substance that can increase the rate of reaction, that can increase the velocity of reaction, that is known as a catalyst. Now enzymes are nothing but the biocatalyst. So bio means biological system or your human body. Okay, so these are the substances, these are the biocatalysts. They will increase the velocity of reaction, they will increase the rate of reaction in the biological system. Okay, so that is nothing but the enzyme. Enzymes are nothing but the biocatalyst. Their nature is nothing but the protein. They are proteinous macromolecules that catalyzes the biological reactions human body. So they are the biocatalyst, and they are the protein in nature, and they have these specificities. They are specific in their action. Their action is rapid and accurate. So enzymes are biocatalyst. They are protein in nature. They have specific action, rapid action, and accurate action, and they will increase the velocity of or rate of reaction without undergoing any change in themselves in overall process. So enzymes, they will take a part in the reaction, enzyme catalyzed reaction. They will increase the rate of reaction. They will enhance the formation of product themselves. They will not undergo any change in the overall process. That is known as a enzymes. Okay. Now what will they do? What exactly enzyme does? So enzyme lowers the activation energy required for the enzyme catalyzed reaction. What will enzymes do? They will decrease, they will lower the activation energy of activation जो बोलते हैं अपन वो lower कर देंगे, so that reaction will be proceeded faster than normal rate. Okay, so that's why enzymes will decrease the activation energy, they will lower the activation energy, and this was question in GPAT 2022. You can go and check the paper. Okay, so they used to lower the activation energy. Okay, so a lot of students, they got confused that catalyst, they used to lower the activation energy, but how enzymes? But enzymes are nothing but the biocatalyst, so they are also a catalyst present in the biological system. Okay, so obviously they will also decrease the energy of activation, they will lower the energy of activation, and this was question in GPAT 2022 recently. Okay, so enzymes, they will increase the rate of reaction, they will increase the velocity of reaction, they will speed up the reaction without undergoing any change in the overall process. Okay, they will just enhance the velocity of reaction by decreasing the activation energy. Okay, now this there is a little bit history behind these enzymes. So this Buckner in 1883 was isolated one principle known as a zymase from the cells of fungus. So Buckner in 1883 isolated one principle known as zymase from the fungal cell. Okay, and that zymase is nothing but the enzyme, that is the enzyme function. So that is enzyme function isolated from fungal cell in 1883 by Buckner. Now this zymase is responsible for the conversion of sugar into alcohol. Okay, this was again a question in pharmacist exam. So what is the role of your zymase? No, it is a question in GPAT 2020 only itself. Zymase is a enzyme that convert alcohol sugar into alcohol. So natural sugars will be converted into alcohol by this zymase enzyme. Okay, so this was the first enzyme that was isolated. Okay, इसके ऊपर दो क्वेश्चन बन सकते हैं. देखो zymase क्या करता है? Zymase is the enzyme that convert sugars into alcohol. Okay, and it was the first isolated enzyme. Okay, remember Buckner in 1883 isolated the principal zymase which convert the sugar molecule into alcohol. Okay, and which is later found to be a protein in the nature. Okay, ये zymase जो है वो proteinous nature है, उसका protein nature है. So these enzymes are nothing but the protein in nature. They have specific native tertiary structure. उसमें tertiary structure होता है इसमें protein में. You just try to write down few things which are not listed over here in the presentation, but I used to tell you कुछ चीजें हैं कि यहाँ पे सामने नहीं है लेकिन मैं बोल देता हूँ तो वो इम्पोर्टेंट है. जैसे Zymase was the first isolated enzyme, यहाँ पे कहीं लिखा नहीं लेकिन मैंने बोल दिया. Buckner in 1883 isolated one principle from fungus cell suspension known as a zymase. Okay, it is a protein in nature and this was the first enzyme isolated. It convert alcohol into sugar. Okay, then again James Summer first achieved the isolation and crystallization of urease. Then very after this Buckner, James Summer also isolated one enzyme known as a urease. Urease means the enzyme responsible for the breakdown of urea. That was isolated from Jack Bean. So this is just a important historical background. बहुत सारा है इसमें बुक्स में काफी एक पूरा पैराग्राफ दिया होता है इसके ऊपर हिस्ट्री के ऊपर, but everything is not important for our point of view. So what is important for us that I have been listed over here. Now next is a nomenclature of these enzymes. So there are different ways to know to give names for this enzyme. So depending upon the nature of substrate कि एंजाइम का सबस्ट्रेट कौन सा है. जैसे प्रोटीन है, अगर प्रोटीन एंजाइम का सबस्ट्रेट है सो दैट इज नथिंग बट द प्रोटियोलाइटिक एंजाइम. फॉर एग्जांपल ट्रिप्सिन, कीमोट्रिप्सिन, ओके, पेप्सिन, those enzymes are found in the stomach है ना. आप those enzymes are responsible for the digestion of protein in the stomach. So those pepsin, trypsin, chymotrypsin as enzymes may be classified into enzymes may be named into different ways. So enzymes may be named depending upon their substrate. ये एंजाइम के सबस्ट्रेट कौन सा है, सबस्ट्रेट कौन सा है उसके ऊपर भी एंजाइम का नॉमेनक्लेचर होता है. जैसे बोला है मैंने आपको कि proteolytic enzymes. So substrate is nothing but the protein for themselves. Okay. So pepsin is there, trypsin is there, chymotrypsin. So they will degrade the protein. That's why they have given the names. Again, then ASE. ASE is the suffix. Most of the enzymes they having the suffix of ASE. For example, urease. So ASE is zymase. ASE is the glucase. ASE is, so they will provide some kind of information. Urease means that will break down the urea. Zymase means that will convert glucose to sugar. Okay. Protease means that will convert break down the protein. So likewise that. So these kind of names they can be given to enzyme. Okay. Again another way to naming the enzyme is the depending on the name of discoverer. So जिसने discover किया उसका नाम दे दो. That is the name. But these names will not provide you the accurate idea that what is the total picture, what is the total scenario, they are not going to tell you. So that's why these names are known as a trivial names. These names are the trivial names. Now for getting a proper idea कि हर एक एंजाइम क्या करता है एग्जैक्टली, कौन सा रिएक्शन कैटलाइज करता है, कि वो सबके बारे में इंफॉर्मेशन गेन हो जाए, ऐसे एक नोमेनक्लेचर का सिस्टम है. So International Union of Biochemistry, International Union of Biochemistry, they have given a nomenclature for these enzymes. They have given a names to the enzymes. They have categorized all the enzymes into 8 categories. 8 categories में उन्होंने उनको डिवाइड किया है और उनका nomenclature किया है. जैसे oxidorreductase, hydrolysis, ligases, ligases, isomerases, likewise that. We are going to see that. Don't worry. That is known as the International Union of Biochemistry nomenclature. Okay, and each and every enzyme has given a 3-digit code. Each an enzyme has given a 3-digit code. Okay, so sorry, 4-digit code. So first digit will describe the class of enzyme, second digit will describe the subclass, third digit will describe the actual enzyme. So likewise that. Okay, now these enzymes may be either intracellular or extracellular. ठीक है, so सेल के अंदर जो enzymes है, so that is known as an intracellular enzymes. सेल के बाहर जो एंजाइम्स है, that is known as extra cellular enzymes. So intracellular enzymes, they are present inside the cell, they are produced inside the cell, and they will function, they will perform their specific function inside the cell only. They are known as intracellular enzymes. Okay, for example, enzymes responsible for the replication, transcription, translation, they are present inside the cell. That's why they are known as intracellular enzymes. Now extracellular enzymes means what? They are present in the, they are not present inside the cell, they are inside the cell, and they are secreted over there. They are known as extracellular enzyme. For example, all digestive enzymes. Okay, जितने भी digestive enzymes होते हैं आपके, जैसे lipase हो गया, amylase हो गया, amylase, lipase, protease, trypsin, chymotrypsin. So all these enzymes are the extracellular enzymes. Okay, depending on their function. Okay, depending on their function also, enzymes may be classified. So lipase, for example, lipase means that will break down the lipids. Glucose means that will break down the glucose. Likewise that. Now again, now whatever the enzyme is, so that enzyme is made up of different parts. Okay, so active enzyme is known as a hollow enzyme. Active enzyme is known as a hollow enzyme. Okay, and hollow enzyme is made up of two parts. Okay, first one is known as a protein part, and second one is known as a non-protein part. So protein part is nothing but the apoenzyme. Now enzyme is made up of a active enzyme is known as a hollow enzyme. Okay, now it is made up of two parts. So first part is nothing but the apoenzyme. First part is nothing but the apoenzyme, and second part is nothing but the coenzyme. So apoenzyme is nothing but the protein part of your enzyme. Okay, protein part of your enzyme is known as a apoenzyme, and non-protein part of your enzyme is known as a coenzyme. Okay, so ये बहुत बार पूछा है इसके बारे में. Okay, so active enzyme consist of, और आपको नीचे देंगे hollow enzyme, apoenzyme, coenzyme. So hollow enzyme is known as an active enzyme. Enzyme itself is made up of two parts, protein part and non-protein part. Protein part of enzyme is known as a apoenzyme. Okay, इसके ऊपर भी एक दो बार क्वेश्चन आ चुका है. So apoenzyme is a protein part and coenzyme is a non-protein. So apoenzyme protein part, coenzyme non-protein part. Okay, now let's see. आपको अगर क्वेश्चन आया, so which part of the enzyme is responsible for the enzyme specificity? So enzymes are very specific in their nature. They are having substrate specificity. They are having reaction specificity. They are having optical specificity. D-glucose. D-glucose will only catalyze the conversion of D-glucose. Lactate dehydrogenase. L-amino aminase. So that will only catalyze the L-amino acid reaction, not D-amino acid. So they are having structural specificity. They are having substrate. They are having optical specificity, and this specificity of the enzyme is because of the protein part. Okay, and that is nothing but the apoenzyme. So if they ask any question like that, like for example, optical specificity of enzyme is because of the one of the following part of, and they will give you option like hollow enzyme, like apoenzyme, coenzyme, cofactor, or prosthetic group. So apoenzyme will be the answer because that is a protein part, and protein part of enzyme will only determine the specificity, will only responsible for the specificity of your enzyme. Remember this. Now coenzyme is non-protein part. Okay, coenzyme is non-protein part. So most of the coenzymes, they are made from vitamins. Okay, like NAD, NADPH, etc. So most of the enzymes, they require certain coenzymes for to be effectively active in the reaction. They require a coenzyme, which is a non-protein part. Now if this coenzyme part, if it is loosely attached to a apoenzyme, अगर protein part and non-protein part अगर loose है, so if non-protein part, protein part of enzyme, they are attached by loose bonds, they are known as a coenzyme. And if that is attached by relatively stronger bonds, then that part or that coenzyme is known as a prosthetic group. The term prosthetic group, okay, the term prosthetic group is used when non-protein part that binds covalently with the apo enzyme. ठीक है, नॉर्मली क्या होता है, ये कोई एंजाइम जो है apo एंजाइम को लूज बाइंड करता है, जैसे आपका कोऑर्डिनेट बॉन्ड वगैरह. सो इन दैट केस if non-protein part of your enzyme is bound to apo enzyme or protein part by loose bond, that is known as a coenzyme, and if that non-protein part binds to the protein part by covalent bond, that is known as the prosthetic group. Okay, again this is very important, इसके ऊपर कम से कम 3 या 4 बार क्वेश्चन आ चुका है, कंफ्यूज नहीं होना है. Okay, hollow enzyme is known as a active enzyme. Okay, it is made up of two part, apo enzyme and coenzyme. Apo enzyme is protein part which will also determine the specificity of enzyme. Okay, coenzyme is a non-protein part and it is loosely bound to a protein part. If it is tightly bound to protein part, then it is known as a prosthetic group. Please mute yourself. Then functional unit of enzyme is known as a hollow enzyme. So I have told you functional unit of enzyme is known as a hollow enzyme. Now one enzyme will have multiple forms. Okay, like they are known as isoenzymes. जैसे एक ही एंजाइम का डिफरेंट डिफरेंट फॉर्म्स होगा, उसको बोलेंगे आइसो एंजाइम. जैसे साइटोक्रोम P450 that is to metabolize the most of the drugs. साइटोक्रोम P450 that is known as माइक्रोसोमल एंजाइम्स. ओके, तो उसमें एक ही एंजाइम है क्या, उसमें बहुत सारे CYP2A34 CYP P2T, ऐसा बहुत सारे, देयर आर सब फैमिली ऑफ द साइटोक्रोम P450. They are known as iso enzymes. Okay, so multiple forms of same enzyme, they are known as isoenzymes. Then now we'll see the next point. So now if enzyme is made up of a single polypeptide, so enzymes are nothing but they are a protein in nature, they are a macromolecule protein in nature होता है, tertiary structure होता है उसका. So enzymes will have, they are proteinous in nature and they will have tertiary structure. Okay, tertiary structure. Now if enzyme is made up of one single polypeptide से अगर enzyme बना है तो उसको बोलेंगे मोनोमेरिक एंजाइम. मोनोमेरिक एंजाइम. अगर एंजाइम अगर दो एक से ज्यादा पेप्टाइड से बना है तो उसको बोलेंगे ओलिगोमेरिक एंजाइम. ओलिगोमेरिक एंजाइम. अगर एंजाइम अगर दो या तीन एंजाइम साथ में मिलके एक एंजाइम बनाते हैं तो उसको बोलेंगे मल्टी एंजाइम कॉम्प्लेक्सेस. I will explain this again. Remember, now enzymes may be either monomeric. Monomeric means they are made up of a single polypeptide. Okay, single polypeptide. For example, ribonucleases and trypsin. So these enzymes, they are made up of a single polypeptide and they are called as a monomeric enzyme. Now oligo means few. Oligomeric enzymes means they are made up of a few polypeptide. For example, lactate dehydrogenase and aspartate transcarboxymylase. So these two enzymes, they are known as oligomeric enzymes because they are made up from few peptides. Okay. Now multi-enzyme complexes. Multi-enzyme complexes means what? So if two or more enzymes, they are joining together to perform a specific function step by step, they are known as a multi-enzyme complexes. For example, pyruvate dehydrogenase and fatty acid synthetase. These are the examples of multi-enzyme complexes. So remember what is enzyme, what is the mechanism of action of enzyme. GPAT 2020 में ये भी क्वेश्चन था. उसके बाद में GPAT 2020 में सॉरी ये क्वेश्चन था जाइमेज का. Zymase will convert glucose into sugar. Okay, and it is a first isolated enzyme. Remember इसके बारे में क्वेश्चन आ चुका है. Then again इसके बारे में दो बार क्वेश्चन आ चुका है, दो तीन बार. So hollow enzyme is an active unit of enzyme made up of two parts, apo enzyme and coenzyme. Apo enzyme is a protein part and coenzyme is a non-protein. Protein part will determine the specificity of enzyme. So optical specificity और substrate specificity, कौन सा substrate के ऊपर act करना है उसको, या फिर reaction specificity, that is determined by this apo enzyme part. Okay, then functional unit of enzyme is known as a hollow enzyme. Remember, then oligomeric enzyme, monomeric and multi enzyme complex. I have told you. Now next part is, as I have told you, जैसे कि मैंने बोला है आपको कि enzymes may be classified into different ways. जैसे trivial names. So I have told you like depending upon the substrate. अगर substrate कौन सा है, जैसे अगर protein substrate है enzyme के लिए, so those for example pepsin, trypsin, chymotrypsin, they have named depending upon their substrate. जैसे chymotrypsin, trypsin and pepsin. So all these three enzymes, they will act on a protein only. So proteolytic enzymes. You know? So then depending on the substrate like glucose, so glucose will act on a glucose like that. Lipase will act on a lipid. So likewise that depending on the substrate they have given the name, and they are also given the name on the disc on the basis of discovery. So those are the trivial names, but those names will not give you more information how there is a mechanism of enzyme, what kind of reaction is catalyzed. So that's why International Union of Biochemistry, they have classified enzyme in these 6 categories. And what are these categories? These are nothing but the oxidoreductase, then transferases, then hydrolyses, liases, isomerases, and ligases. Now these are the categories. These are the categories. Now इसके ऊपर already GPAT 2021 में क्वेश्चन पूछा है. तो क्वेश्चन क्या था अपन देखते हैं. So oxidoreductase, we'll see it. What are the different enzymes and what are the their names? What are the type of reaction catalyzed by the enzyme or the class of enzyme? Now, first one is a oxidoreductase. First one is a oxidoreductase. As the name indicates, oxidoreductase means what? So they are involved in the oxidation reduction reactions. So oxidoreductase means they are involved in the oxidation reduction reactions. Oxidoreductase, oxidation reduction reaction. So for example, alcohol dehydrogenase, cytochrome oxidase, okay, then amino acid oxidase. These are the enzymes, they are involved in the oxidation reduction reaction. So type of reaction catalyzed, कौन सा है? Oxidation reduction. So what do you mean by oxidation and reduction? Gaining and losing of electron respectively. Oxidation means gaining of electron, reduction means losing of electron. This was question in GPAT 2021. ऐसा क्वेश्चन था. So one of the following category of enzymes are involved in the reaction which are involving gaining and losing of electron. The reaction which are involved in gaining and losing of electrons. So oxidoreductase, these are the enzyme involved in the oxidation reduction reaction. तो ऑक्सीडेशन मतलब क्या है? गेनिंग ऑफ इलेक्ट्रॉन. रिडक्शन मतलब क्या है? लूजिंग ऑफ इलेक्ट्रॉन. ठीक है. सो दोज category of enzyme which are involved in the gaining and losing of electrons in the reaction. So that is the oxidoreductase. ये ऊपर ऐसा ही क्वेश्चन था जीपीएट 21 में. You can go and check the papers. Now second category of enzymes, that is known as the transferases. Transferases, as the name indicates, transferases. As the name indicates, transferases means क्या? Transfer of what? Transfer of groups. So they are involved in the transfer of groups. So example is hexokinase, transaminase, transmethylase, and phosphorylase. So hexokinase means आपने देखा होगा, you might have seen in the biochemistry of carbohydrates, metabolism of carbohydrates. In that, in glycolysis, very first enzyme, that is a hexokinase, that will convert glucose into glucose 6 phosphate. Okay, so 6 phosphate मतलब क्या? 6 position पे phosphate group आ गए. So hexokinase is involved in the transfer of group. So it will transfer phosphate group to glucose. So hexokinase, transaminase means it will transfer the amino group. Transmethylase, it will transfer the methyl group. Phosphorylase, it will transfer the phosphate group again. Okay. So these category transferases, they will transfer one group from one to another molecule. Okay. Then third category is nothing but the hydrolysis. Hydrolysis. So as the name indicates, they are involved in the hydrolysis process. Okay. So example is lipase. Lipase means it will break down, it will cause hydrolysis of lipids. Cholinesterase means it will break down the acetylcholine molecule. It will hydrolyze the acetylcholine molecule into acetyl group and choline and acetic acid. Okay, so acetylcholine esterase or choline esterase, that will hydrolyze the acetylcholine molecule into choline plus acetic acid. Then alkaline phosphatase is also hydrolyzed enzyme. Pepsin, urease. Urease के बारे में बोला है. Urease means it will break down the urea. So hydrolysis category of enzyme, they are involved in the is hydrolysis or breakdown of. Now next one is a liases. Next one is a liases. So these category of enzymes, they are involved in the addition or elimination of groups. Addition or elimination of groups. So addition or elimination of groups. For example, addition of hydroxyl group or elimination of hydroxyl group. So liases. So उसका एग्जांपल है अल्डोलेज, फ्यूमरेज एंड हिस्टिडेज. टाइम पे आते जाओ. क्या होता है? आधा चैप्टर हो चुका है अभी. नेक्स्ट वन इज, next category of enzyme is nothing but the liases. Liases means they are involved in the addition or elimination reactions. So for example, aldolase, fumarase, and histidase. So they are involved in the addition and elimination. addition of hydroxyl group, addition of amino group, addition of carbon dioxide. Okay, these are known as liases. Next one is isomerase. Okay, isomerase means what? They will they will cause isomerization reaction. For example, phosphoexose isomerase. You might have seen in the glycolysis, the third step in the glycolysis. Okay, glucose 6 phosphate is converted to fructose 6 phosphate by the isomerase enzyme. Okay, you might have seen in the glycolysis. So that's isomerization reaction is catalyzed by a kind of enzyme, category of enzyme known as a isomerase. So phosphoexose isomerase, then glucose isomerase, etc. They are involved in the isomerization reaction. Okay. Then next category of your enzyme is nothing but the ligases. Ligases means to bind. Ligases means to bind or to synthesize. To bind or to synthesize. So they are involved in the condensation reactions, and they are mostly usually or they are mostly ATP dependent. So those reaction catalyzed by ligases enzyme, ligase or ligase enzyme. The ligase means to join. Join किसको करेंगे? Ligase are the enzymes. They are involved in the condensation reactions, and they are usually ATP dependent. For example, glutamine synthesis. That is succinate thiokinase. So these are the enzymes. Those are known as a liases, ligases. Now, next point is nothing but the okay factors affecting on the enzyme activity. So this point is again important, and इसके ऊपर भी question आ चुका है. So we'll see that factors affecting the enzyme activity. Now, enzyme activity can be affected by numerous factors. For example, the concentration of enzyme, concentration of substrate, the concentration of reaction product, okay, then the temperature, then the pH, etc., etc., inhibitors and modulators, etc., etc. There are numerous factors which can affect the enzyme catalyzed reaction. Suppose that you have one enzyme catalyzed reaction, आपके आपके पास क्या होगा? आपके पास रिएक्शन मतलब रिएक्टेंट होगे, एंजाइम होगा. सो जो रिएक्शन है उसको कौन अफेक्ट करेगा? फर्स्ट, concentration of enzyme. If the concentration of enzyme is increased, the velocity of reaction will be also be increased proportionally. So proportionally increase in the velocity of reaction. If you increase the concentration of enzyme, so reaction velocity will also increase proportionally. Then next factor is nothing but the concentration of substrate. So increase in the velocity of reaction within a certain range, okay, within a certain range of substrate level. तो अगर आपने सबस्ट्रेट का कॉन्सेंट्रेशन इंक्रीज किया लेकिन एंजाइम का नहीं, सिर्फ सबस्ट्रेट का, तो कुछ लिमिटेड रेंज तक रिएक्शन जो है, वेलोसिटी जो है रिएक्शन का इंक्रीज होगा और बाद में क्या होगा? कांस्टेंट रहेगा. So increase in the velocity of or rate of reaction is there when increase in the substrate concentration at a certain range, and after that there will be constant. So एक hyperbola type का एक आपको curve मिलेगा. So that is a concentration. Now order of reaction. So first order with respect to substrate. Order of reaction. Now order of reaction means reaction order. Reaction कितने factors के ऊपर depend है? Reaction rate of reaction depends on only one factor, that is known as a first order reaction. If rate of reaction depends on the two factors, that is known as a second order reaction. If rate of reaction, ठीक है, first order, second order, and mixed order. Mixed order मतलब more than two factors. So first order means rate of reaction will depend on the one factor only. So velocity of reaction is almost proportional to a substrate concentration when substrate concentration is less than Km. What is Km? Km is nothing but the amount of enzyme required to produce half of maximum velocity. Km is nothing but the Michael is maintained constant. It is nothing but the amount or the concentration of enzyme required to produce half of maximum velocity in the enzyme catalyzed reaction. In the first order, velocity of reaction is almost proportional to the substrate concentration if substrate concentration is less than Km. Now, if substrate concentration is much greater or it is higher than Km, reaction is independent of substrate, and now it is called as a zero order reaction. Order of reaction may be either zero order, first order, second order, or mixed order. तो अगर आपका substrate concentration Km से कम है, मतलब Km is a Michael is maintained constant, ये क्या है बाद में देख लेंगे. If substrate concentration is less than Km, then order of reaction will be first order. If the substrate concentration is greater than Km, then order of reaction will be zero order. Zero order means the rate of reaction will not be dependent on any factor. Now suppose that there is one enzyme catalyzed reaction. Suppose that there is an enzyme catalyzed reaction, जैसे आपने यहाँ पे देखा है enzyme plus substrate will form enzyme substrate complex. Enzyme substrate complex, and this enzyme substrate complex will be then converted into product. Okay, product and enzyme will be free. So enzyme will not undergo any changes in the reaction. Enzyme will be free. You will get your product. This is a reaction. Now consider enzyme plus substrate. Okay, forward reaction का constant है आपका K1. This is a constant for forward reaction. K2 is a constant for backward reaction because this reaction is in equilibrium. Now enzyme substrate complex form हो गया आपका. तो ये किसके ऊपर डिपेंड है? देयर आर टू कांस्टेंट K1 and K2. So formation of enzyme substrate complex depends on two constant, forward reaction constant and backward reaction constant K1 K2. And then enzyme substrate complex is finally converted into product by K3. K3 is the forward reaction constant. Now Km means what? Km is known as a Michaelis Menten constant. Km is known as a Michaelis Menten constant, or it is also known as a Briggs-Haldane constant. Briggs-Haldane constant or Michaelis Menten constant. Km is equal to K1 plus K3 divided by K1. K1 plus K3 divided by K1. K, that is a K means constant at rate of reaction, the velocity of reaction. ठीक है, जैसे एंजाइम प्लस सबस्ट्रेट फॉरवर्ड रिएक्शन में एंजाइम सब कॉम्प्लेक्स होता है तो उसका एक वेलोसिटी होगा ना रिएक्शन का, इज नोन एज K1. एंजाइम सबस्ट्रेट कॉम्प्लेक्स जो बना है दैट इज अ रिवर्सिबल प्रोसेस, तो बैकवर्ड भी रिएक्शन हो सकता है. एंजाइम सबस्ट्रेट कॉन्सेंट्रेशन का डिसोसिएशन होके एंजाइम एंड सबस्ट्रेट आपको अलग अलग मिल सकते हैं. तो बैकवर्ड रिएक्शन का जो कांस्टेंट है दैट इज K2. एंड एंजाइम सबस्ट्रेट जो है वो प्रोडक्ट बना सकता है तो उसका रिएक्शन कांस्टेंट है K3. This Michaelis Menten constant, Briggs and Haldane's constant is nothing but the reaction constant. Km is equal to K2 plus K3 divided by K1. So final जो order of final जो velocity of reaction है वो कैसे calculate constant? So that is known as a Michaelis Menten constant. So Michaelis Menten equation. So V is equal to V max into substrate concentration divided by Km plus S. So V मतलब final velocity of enzyme catalyzed reaction. Max means maximum velocity. S is nothing but the substrate concentration. Km is nothing but the Michaelis Menten concentration. Michaelis Menten constant, and S is nothing but the again substrate concentration. Another way you can see V is equal to V max into substrate concentration divided by Km plus that is substrate concentration. This is known as the Michaelis Menten equation. So V is the measured velocity, V max is the maximum velocity, S is the substrate concentration, and Km is the Michaelis Menten concentration. What is Km? Michaelis Menten concentration, and it is nothing but the concentration, substrate concentration to produce a half of maximum velocity. Substrate concentration to produce a half of maximum velocity. You plot अगर आपने या फिर आपने ग्राफ ड्रॉप किया, so if you plot a substrate concentration on x-axis and velocity on y-axis, तो आपको ग्राफ मिलेगा. ठीक है, ग्राफ मिलेगा. So initially आपका इंक्रीज हो जाएगा, बाद में कर्व आएगा और बाद में फ्लैट रीजन. If you intercept y and x at a point, you will get a this point. Now the substrate concentration which is required to produce half of maximum velocity is known as a Km. So this is the Michaelis Menten equation. ये याद रखो, Michaelis Menten equation के बारे में भी क्वेश्चन आ चुके हैं. V is equal to V max into S divided by Km plus S. This is the Michaelis Menten equation. So that is nothing but the reaction kinetics or enzyme kinetics. Enzyme kinetics are described by different processes like zero order and mixed order. Mixed order kinetics, they are described by Michaelis Menten equation. एक याद रखो क्वेश्चन, इसके ऊपर भी क्वेश्चन. Next point we'll see. Now we have seen the Km value. So Km value is nothing but it is a constant characteristic feature of the particular enzyme. Now at a low Km value,